So we have been very active in the MINDACT and Transmit Consortium for 10 years right now, and active in the pre-MINDACT setting and then in the MINDACT trial. So. And this is why I was so proud to present the results. There are no new results today. The paper will be released. But well, it was the occasion to rethink about it because this is a complicated trial, and the application of the results are very important, but still a bit discussed of how do we choose the prescription. So the primary endpoint was to verify that using the genomic test in patients who are classified as clinical high risk allowed to avoid chemotherapy safely. Meaning that we expected that in this population we would have 95% distant metastasis-free survival at 5 years, and we allowed ourselves a confidence interval with the lower bound being upper to 92%. We consider that lower than 92 wouldn't be acceptable, and this is a very conservative cutoff indeed, but well, it was very important because if it's lower than 92%, there would be an important risk that chemotherapy benefit would be higher. So indeed, MINDACT reached its primary endpoint, which is the lower bound of the confidence interval is 92.7. Yeah. But the DMFS at 5 years is 94.7, so very high in this high risk population, and almost half of them being node positive. So this is very relevant population for which we have the question every day and for which we still mostly prescribed chemo until today. So this is going to be very relevant. We just need the reimbursement in most countries, but I think provided those results, we could change and provide the patients with a genomic test to try and avoid chemo among this category. So almost 50% of them avoid chemotherapy. Well, it was exactly as expected regarding clinical low genomic low, which is very high DMFS rate at 5 years, almost 98%. So in this population, there is no way anybody would prescribe chemo. For the clinical high genomic high, those patients have received chemo, and we are at 90% at 5 years. So this is very relevant, telling us that we need to do much better for this population. So it's good to show that they need chemo and that, well, they probably benefit from chemo. We couldn't show it in MINDACT, but from previous results. But they need additional treatments or replacing treatments instead of chemo because it's still not perfect. 90% is not bad, but it's not at all perfect. And well, very interestingly, the two groups, clinical high genomic low and clinical low genomic high, did almost the same, around 95% at 5 years DMFS. And well, this is, well, it's especially questioning regarding the clinical low genomic high. Well, indeed, we didn't know really what to expect. This population is clinically very different. It's mostly node negative, well, kind of good tumors. And well, it's interesting that it finally did almost the same. Some of them received chemo but most of them did not. Well, it's a small population, around 500 overall, and this population, it does not appear that there is any benefit from chemo, but well, the trial is not powered for that. So, well, I don't think it's useful to use genomic the genomic assessment for clinical low population, because at the end you don't know what to do with it. So right now we would stick with clinical evaluation, and that's it for this population. Yeah. For MINDACT, there are some upcoming results, but the main paper will be very detailed in terms of subgroups and everything. But the main results will be the secondary randomization. So at San Antonio, we'll be presenting the chemo randomization. It's going to be published next year. So it was for all patients who received chemo, we proposed a randomization between standard sequential anthracycline-based treatment and taxanes for the node positive. And the other arm was docetaxel plus capecitabine, which is a very relevant question these days because the role of capecitabine has been questioned a lot recently. So, well, I don't know anything about the results, I can tell you. And we will have all of this at the end of the year. And the secondary endpoint was a randomization regarding endocrine therapy, but it's not for now. It's going to be in well, almost 5 years. So, but the most interesting results probably are the next results regarding biological assessments and complete sequencing of the whole bench of tumors and assessing prognosis to sequencing to other ways of, well, besides transcriptome. So this is going to be very interesting, but this is exploratory. I mean, the main endpoints are here. The other ones are totally exploratory. It's more science, but it should provide us with a lot of information. I think it's always very important to mention that we won't be able to do such trials anymore again because it costs almost 50 million euros overall. And it was a huge effort just to reach the level 1A biomarker. So we just need another way to reach the level 1A. It's almost impossible. And I don't think medicine and science can expect 10 years before reimbursing genomic tests, whereas finally the retrospective studies told the same thing. So I think we have to, well, a bit more modern regarding statistical tests and the way we assess biomarkers. If we have clean, analytical studies, etc. So this is very important.